In this class, we are going to study about the first category of the kingdom fungi, and that is the lower fungi. And the normal name for this lower fungi is Phycomycetes. So this Phycomycetes is one of the primitive type of fungi, and therefore it has certain properties of the mycelium which are different from the higher fungi like Ascomycetes and Basidiomycetes. So first of all, we have to see the habitat of this Phycomycetes, that is where it is found. So the first and most important feature of its habitat is the availability of water or moisture. So these are normally found in areas of either aquatic environment or very moist condition along with warmth or heat. So these are normally found in aquatic environment. Apart from that, these are also present on decaying plant matter. So we can say that these Phycomycetes members are saprophytic, that is these decomposed decaying plants, and therefore these can be termed as a decomposer. So these are also found on decaying plant matter. Apart from that, these are also present in very moist and warm conditions like warm and moist soil of the summer season. So this is the normal habitat of the Phycomycetes. So these are aquatic or on decaying wood, that is these are saprophytic. Apart from that, these occur in moist and warm conditions, and these organisms, apart from being decomposers or saprophytic, these are also parasitic. That is, these cause diseases in various organisms like plants and certain other species also. So here we will later study about the parasite species or the parasitic species of the Phycomycetes. These are the examples of those parasitic species. So the parasitic species obviously reside on or inside a plant, that is these are normally plant parasites. Very few are also the animal parasites, but mostly we have to study about the plant parasites over here. So this was about the habitat or the place where the Phycomycetes are found. Apart from that, we know that the body of a fungi is normally composed of mycelium, and the mycelium is formed by a network of thread-like structures which are almost translucent in nature, and that is called as hypha. So in this case, the hypha does not have cross walls or cross boundaries or cell walls between two distinct cells. Therefore, the hypha or the mycelium over here is aseptate, that is cross walls or septum does not occur between the cells. So that is why these are called as aseptate. Apart from that, when the cell wall are absent or walls are absent between the cells, therefore cytoplasm of various cells are intermingled with each other, and therefore it is called as multinucleate or cenocetic. So such type of hypha is called as cenocetic and also as multinucleate. Apart from that, this hypha are branched. So branched hypha is also a very important feature of Phycomycetes, and in some cases we will observe that branched hypha will not be present, but in Phycomycetes branched hypha is a very common occurrence. So this was about the habitat as well as the mycelial properties of the Phycomycetes, which will be general in almost all the species of Phycomycetes. Now in Phycomycetes, we have to study about two more categories, and those are called as Uvogamus and also the Zygomycetes. So the name of the Zoospore or the Uvogamus is dependent on the spore in which that is being formed in that case. For example, in Uvogamus we have the formation of oospore, but in Zygomycetes we have formation of zygospore. So on the basis of the spore that is being formed by the sexual reproduction, the category has been named. So the Zygomycetes has been named after zygospore, and the Uvogamus has been named after the oospore. So that is why, that is the relation between the name and the type of sexual reproduction product that are being formed. So these are the two categories that we have to study under Phycomycetes. Now we first start from the presence of substances in the cell wall. So we normally, when we talk about fungi, we hear a word and that is called as chitin in the cell walls of the fungi. So here in case of Uvomycetes, we observe that chitin is almost absent and the main material by which the cell wall has been formed is cellulose. So it is similar to the plant. So here the main cell wall material is cellulose and chitin is almost negligible. Apart from that, some other glucans. So these glucans are also some polysaccharides which are formed by glucose, and in glucose we have various forms of glucose and these glucans are formed by D-glucose. So you will study more about D-glucose in the chapter biomolecules. So these glucans are also a polymer or polysaccharide which are formed by D-glucose. But in the case of Zygomycetes, we observe that the cell wall is primarily composed of the substance that we are very familiar with in the kingdom fungi, and that is the chitin. So chitin is commonly called as fungal cellulose. So this chitin is also a polysaccharide definitely, but still it has some properties different from the cellulose, but both of these are polysaccharides. So this was the first difference in the cell wall of these two categories. Now the second difference is between the asexual production or asexual reproduction. So in both these categories you can observe that sporangia are formed. So sporangia are those structures inside which the spores are formed. So here also we have the formation of sporangia and here also we have the formation of sporangia. But the main difference is that the sporangia here is behaving like a dual property substance or dual property structure. So when the environment is aquatic or the water availability to the fungi is pretty much, in that case this sporangia will form something called as zoospore. And in our previous classes we have already studied about the various zoospores. So these zoospores have flagella in them and that is why these are motile. So in aquatic conditions the sporangia will produce the zoospores. But if the conditions are dry, or we can say that the condition is terrestrial or the habitat is terrestrial, so in that case these sporangia will behave as conidia. So here the same structure could give rise to two types of spore. One is the zoospore and the other is the conidia. So two types of spores are observed over here. But in that case we have observed the presence of some other type of spore. So in that case the sporangia will produce something called as sporangiospore. Now the sporangiospores are mostly non-motile, but these can also be motile. So in this case the sporangia are producing spores which are known as sporangiospores. And apart from that, here the formation of another type of spore which is unique is occurring and that is called as aplanospore. Now before studying about aplanospore, we have to remember that this aplanospore is not only present in fungi. Aplanospore could also occur in various green alga and also in various protists. So this aplanospore is not unique to the kingdom fungi. It is also found in alga and also in protista. So these aplanospores are non-motile spores and these are very resistant spores. So these aplanospores are capable of surviving very harsh environment or unfavorable climatic conditions. But the zoospores which are being formed here and conidia which is being formed here, these are not very resistant spores. So this is the difference between the spores over here. Aplanospore is pretty much resistant, but the zoospores are not resistant to harsh environmental conditions. So this is the difference between the asexual reproduction between the two categories. Now we come to the gametes. What is the property gametes or the sex cells which are being formed? So here we observe that the gametes are non-flagellate. That is, these do not have flagella. So obviously if they don't have flagella, they are non-motile. But in that case, that is under the Zygomycetes, we can see that the gametes which are being formed, these are multinucleate. So the multinucleate structure is called as cenocytic. That is why in that category we have seen there the gametes are called as cenogametes because these are multinucleate. So multinucleate structure is called as ceno. So here also the multinucleate gametes are called as cenogametes. So this was the difference between the gametic property of the two categories. Now the other difference is in the sexual reproduction. So in sexual reproduction we had studied about various types of sexual reproduction, and in that category two were very common, and that was gametangial contact and gametangial copulation. So here we observe gametangial contact and there we observe gametangial copulation or conjugation. So because of the presence of conjugation, these are called as conjugation fungi. So that is the common name of the Zygomycetes. And why? Because of the presence of conjugation. Now conjugation is a phenomenon in which two parents fuse and exchange their nuclei or male pronuclei and then separate to produce daughter individuals. So that is what is conjugation. Now the difference between gametangial contact and the gametangial copulation is that in gametangial contact, the male sex organ, which is called as antheridium, it directly comes in contact with the female sex organ, and that is called as oogonium. So the male sex organ will come in contact with the female sex organ, that is oogonium. So the male sex organ or antheridium will transfer the male gamete or the male nucleus directly to the female sex organ. But in this case, that is the gametangial copulation, the event that will occur is that the structure which will form the gametes, and that is called as gametangia. So here the two gametangia, that is a male or the female gametangia, will fuse together and they will form a permanent structure, that is these will permanently fuse with each other and therefore perform sexual reproduction. So that is the difference between gametangial copulation and gametangial contact. So that is the difference. Now we have another thing which we studied earlier. Here we have a oospore formation and there we have a zygospore formation. So the name of the category, that is Oomycetes, is dependent on oospore, and the name of the Zygomycetes is dependent on this, that is the zygospore. Apart from that, we can see here is that it is called as Uvogamus fungi. Now the reason behind that it is called as Uvogamus, because the Uvogamus, that is one female gamete is formed which is large and which is non-motile. We can see here that gametes are non-flagellate, that means non-motile. So the male and female gametes which will be formed here are non-motile, and therefore one female gamete is large and non-motile. Such type of occurrence is called as oovogamy. That is why these are called as Uvogamus fungi or Uvogamus category, that is the Oomycetes. Now here we have this zygospore. The difference between the zygospore and oospore is that zygospore is a resting diploid spore. It is a resting spore. So that means that this zygospore will not form a new fungi immediately. There will be an intermediate structure or intervening stage after which the fungi will produce a certain mycelium or the main fungal body. So this was the difference between the two categories of PHYCOMYCETES, that is the Oomycetes and Zygomycetes. So instead of studying each of their separate categories or each of their properties separately, when we compare their properties along with each other side by side, so we can easily understand their features as well as their differences. So that was about the properties. Now we come to the examples. In the first line, we have observed some parasites, and these are the parasites that are normally found in the kingdom or the category PHYCOMYCETES. The first one is the Phytophthora infestans. So it causes late blight of potato. So in this disease there is damage of leaf tissue, and therefore the leaf will seem to have many holes, and the main surface of the leaf is damaged. Apart from that, potato, it can also cause disease in tomato sometimes, but mostly in potato. It causes disease mostly in potato. And not just the leaves are infected. Sometimes the potato tuber that we eat, that is also infected. So that is what is occurring by the Phytophthora infestans. This is a parasite. Now we come to the Albugo candida or Cystopus candidus. These are the same name of the same species. So both the names mean the same species, that is Albugo candida or the Cystopus candidus. So these cause white rust of crucifers. Now this crucifer is the category in which we have observed certain categories, that is cabbage or cauliflower or kale or broccoli. So all those vegetables come under this category. So here we have a white rust disease of crucifers. Now we have Pythium and Debeриanum. So it causes damping of disease in certain species, most commonly that is the vegetables and tobacco. So this damping of disease means that a plant will normally grow above the soil, and at the base of the stem will get damaged, and therefore the plant will fall. So that is what is called as damping of disease in tobacco and commonly vegetables that we eat. Apart from that, we have Sclerospora graminicola. So it is a very common disease in certain species like bajra, and in bajra it causes something called as green ear disease. Apart from that, it causes something called as mildew disease also in certain other species like cereals like rice and wheat. So these are also disease causing agent in rice plant as well as wheat plant. And the last and least important is the Saprolegnia parasitica. So there is a fish which is called as salmon, and salmon is a very common fish eaten in many parts of the world. So in salmon fish, it damages the skin of the fish and also the gills of the fish. So when the gills of the fish are damaged, then the gills of the fish will get infected by many bacteria, and therefore the fish will die. So these are some of the parasitic species. So the first four that we have studied, those were plant parasites, and the fifth one, that is the last one that we are seeing, that is an animal parasite, that is in the fish. So it causes disease most commonly in salmon fish, and the first four are causing diseases in plants. Now we come to its categories, and these are very simple, and a very common example that we all know from our childhood is the Rhizopus, which we normally call as the bread mold. That is what we observe on the normal bread after it is kept for a few days. So a Rhizopus stolonifer is one such Rhizopus species which is called as black bread mold. Apart from that, we also have mucor. So in mucus we have Mucor mucido. Apart from that, other species of mucor are also there. So that is called as dung parasite, or we can say that it is Coprophylus. So Coprophylus means that something which grows on animal fecal matter or dung. Apart from that, we also have Pylobilus crystallinus. So this is also a Coprophylus species, that is mucor, and this Pylobolus also grows on dung, that is various animals fecal matter. So these are called as Coprophylus. Apart from Phycomycetes, there are various other fungal species, like the next species that we are going to study, or the next category that we are going to study, is the Ascomycetes. So in Ascomycetes, we will also see there are a very big group of fungi which is Coprophylus, that is it grows on dung or fecal matter of other animals. Now this Rhizopus and mucor are very notorious for us, because these cause spoilage of many of our food stuff like vegetables or fruits or various types of foods that we keep in our kitchen and at various other places. So all those foodstuffs are really threatened by this Rhizopus and Mucor species because these damage our stored foodstuff. Apart from that, some species of Rhizopus and Mucor are beneficial for us because we use them for fermentation, and there are certain foodstuffs in which fermentation is done. So this Rhizopus and Mucor are damaging for us also in terms of food spoilage, but in certain species we have some beneficial uses like fermentation. So these were the most common species, and the most important over here is the Rhizopus, Mucor, the Albugo candida and the Phytophthora infestans. So these four are the most important examples over here. Apart from that, we also have 2-3 extra examples. So focus on these four examples, that is the Rhizopus, Mucor, Cystopus candidus or the Albugo candida. Apart from that, the Phytophthora infestans. So these are the most common examples from here. So now we have completed our first category of fungi, and that is the Phycomycetes. After this, we are going to study about Ascomycetes.